Hello friends, welcome again. Today I am going to make a video on Caenorhabditis elegans, also known as C. elegans. It is a nematode, and in the nematode phylum we all know that we placed only that organisms which are tapeworm like in their structure. So in this diagram you can see the structure of C. elegans, which is somewhat tapeworm like. So it is pointed from the end and somewhat broader in the middle. So this is the structure of C. elegans, and there are many important features present in the C. elegans. That's why we use it as a model organism. It is very useful model organism in the laboratories or for the scientific purposes. So there are some important characteristics present in the C. elegans which makes them as a very important model organism. So what are these characteristics? I will talk about all these characteristics one by one. First characteristic is that that the gene expression of C. elegans can be easily modified. How? So first thing is that C. elegans lives in the soil, and the feeding habit of C. elegans is basically bacteria. So they basically feed upon the bacteria for their feeding, for their food as a food. So in the C. elegans, bacteria is used as a food, and we all know that in the bacteria plasmid are present, and plasmid act as a very important cloning vector in recombinant DNA technology. So we can modify the plasmid of the bacteria, and then we can insert that bacteria directly inside the C. elegans, because the C. elegans feed over the bacteria. So we can easily insert the bacteria inside the C. elegans and easily insert our desired genome by this bacteria. So all another technique like RNA interference technique, which are also used which are also used for as a recombinant DNA technology, for modify the gene expression, or what gene is used as a what gene is expressed, what function, all the things we can know with the help of this RNA interference technique. And here we can directly insert our desired gene into the C. elegans with the help of this bacteria, because C. elegans feed over the bacteria. Second thing is that second feature is that it is hermaphrodite in their structure. Hermaphrodite means both the male and female reproductive systems are present in the C. elegans. So in this diagram you can see these ovary, oviduct, oocytes, and eggs and uterus all represent the female reproductive system, and these blue dots represent the male reproductive system, where sperms are formed. Here eggs are formed. So this is the spermatheca, where sperms are stored. After the fertilization, the eggs comes outside from this opening, which is known as vulva. In the C. elegans, there are basically three openings. One is mouth, one is vulva, and last one is anus. So these three openings are present in the C. elegans structure, and male and female both reproductive systems are present in the C. elegans. That's why it can easily self-fertilize, and it becomes very important model organism. Third thing, that it is very easy to handle. We can easily work with the C. elegans because its structure is very simple. That's why we can use it simply. Fourth thing is that its genome is very homologous to human. We all know that we all know that C. elegans is a eukaryote, and we all are eukaryote also. So there is some similarity of the genome of the C. elegans and the human. So it shows some homologous in their genome, and we all have same ancestor. That's why we show some similarity with the C. elegans. That's why it is a very important model organism. And another thing is that it is the genome sequence of C. elegans is very short or very small. uh, C. elegans have 5 pair of autosomes, and 1 pair is sex chromosome, and the sex chromosome can either be XX or XO. If it is XX, then it is hermaphrodite, because both the male and female reproductive system are present in the structure of C. elegans. And if it is XO, then only one reproductive system is present, usually regarded as male. So XO, if the XO chromosome, sex chromosomes are present, then it is not used as a very important model organism for the scientific purpose. But if it is hermaphrodite in their nature or XX sex chromosomes are present, then it is used as a useful model organism. And this that's why we all use the Caenorhabditis elegans as a model organism, and its genome can be easily manipulated. We can easily manipulate the function of any gene inside the C elegans because their genome size is very small as compared to human. It is eukaryote in their nature, and it shows some similarity with the human chromosomes, and it usually feed upon the bacteria. So all the things are very suitable for C. elegans used as a model organism. One important characteristic of C. elegans is also that its lifespan is very short and it is temperature dependent. How? In this diagram, we can see the lifespan of the C. elegans, and it is dependent over the temperature. In the 15 degrees Celsius, if the temperature is 15 degrees Celsius, then the lifespan of C. elegans is approximately 2.5 days. If it is 20 degrees Celsius, then a lifespan is 3.5 days. And if it is 25 degrees Celsius, the temperature, then the lifespan of the C. elegans can be increased over 5.5 days. So the length of the lifespan is dependent over the temperature. As we increase the temperature, length of the lifespan is also increased. And in the life cycle of the C. elegans, there are some stages, like from the adult eggs are produced, and then these eggs convert into some larval stages, which are represented here by L1, L2, L3 and L4, and after the L4 it convert into the adult. So these are the structures of the lifespan of inside the life cycle of C. elegans. There is one important characteristic of C. elegans. What is this? So here you can see this is a Dauber state. Basically, what is Dauber state? Dauber state is a diapause state, and in this diapause state, C. elegans can live approximately 3 months, which is very important feature of C. elegans. We all know that lifespan of C. elegans is approximately 3.5 days, and it can be increased up to 5.5 days with the increase in temperature. But in this Dauber state, it can remain up to 3 months. So it is very important for C. elegans. In the larval stage of 2, it can goes into the Dauber state. Why it goes into the Dauber state? If there are extreme conditions in the environment, or the environment is very harsh, so in that so to cope with that environment, C. elegans goes into the Dauber state from the larval stage 2. So in this Dauber state, it slows down their metabolism, but not stop. Metabolism continue, but it slows down. And when again the conditions of environment become normal or favorable, then it again convert into the larval forces directly. If there are normal environmental condition, then it goes into the L2 stage, then L3 stage and L4 stage. But when the extreme conditions are present, then the C. elegans goes into the Dauber state from the larval 2 stage, and again when environmental conditions become favorable, then it comes into the direct larval 4 stage. It skips the larval 3 stage. So this is very important feature for C. elegans. That's why we use it as a model organism. And after the larval 4 stage, it again convert into the adult stage, and from there eggs are produced, which can again convert into L4, L1, L2, L3 and L4 stage. So this is about the lifespan of C. elegans, and it is dependent over the temperature. And after the larval stage, after the Dauber stage, when it comes into the L4 stage, then it can approximately live for 15 days, which is also very important feature of C. elegans. So we can increase its lifespan with the help of temperature, and if extreme conditions are present, then it goes into the Dauber state, and then it again comes into the larval 4 stage when the conditions become normal. So this is lifespan of the C. elegans. So all these were the important features of C. elegans. That's why we use as a model organism, because it lives in soil, it feed upon the bacteria, and we can easily modify the gene expression of the C. elegans. It is hermaphrodite in their nature. Its lifespan is dependent over the temperature. Its genome is very very small, so we can easily handle it. All these features makes it very important, according to the point scientific point. So it is very important, model organism of the model organism for the laboratories and for the scientific purposes. I hope this video will help you. Thank you, friends.